Welcome to the e-content learning of Sri Sharda College for Women. My name is Gomati, and this e-content is about mineral nutrition in plants. Now let us get on to the main content. Woodward in the year 1699 observed that plants grow better in muddy water than in clear rainwater. An important observation was made by Saussure in the year 1804 that the minerals present in plant ash are obtained from the soil through the root system. A little after, Liebig in the year 1840 stated that the source of carbon in the plant is carbon dioxide obtained from the atmosphere. Thus, the significance of mineral nutrition in plants is well realized. Now, in this regard, depending on the availability, the quantity of minerals, together with its significance, the elements have been sharply divided into major elements or macronutrients and minor elements or micronutrients. So therefore, getting into all of these, generally, what does all these things do? Now, we shall discuss things in detail. Now, coming to major elements, that is the carbon, hydrogen, and oxygen. These three elements are generally referred to as framework elements and are taken up by the plants in the form of carbon dioxide and water. Virtually all components, excuse me, virtually all components of a living organism contains carbon, hydrogen, and oxygen. So, particularly in the form of carbohydrates, proteins, lipids, and nucleic acids. Some terminate compounds such as rubber are without oxygen. Besides, oxygen is the terminal electron acceptor in respiration. Full respiratory activity can continue even when the concentration of oxygen is just 1%. The pH is finally governed by hydrogen. If there is no carbon dioxide and water on the planet, the plant life shall not exist. Now next, let's move on to nitrogen. It is generally taken up by the plants in the form of nitrates, nitrites, and ammonium salts, and rarely in the molecular form, for example, in the insectivorous plants. It is present in amino acids, which constitute the proteins, in purines and pyrimidines that forms the nucleic acid, coenzyme A, porphyrin, chlorophyll, cytochrome, alkaloids, and vitamins. The deficiency symptom of nitrogen are chlorosis, that is the yellowing which first appears in the older leaves and then in the younger leaves, decrease in protein synthesis, anthocyanin development in leaf petiole, veins, as well as stem, decrease in cell size and cell division. The plants remain stunned or delay or complete suppression of flowering. Now, let us move on to phosphorus. The soil contains phosphorus more in inorganic than the organic form. Plant absorbs it in the form of phosphate ions. The acidic soil has monovalent form, that is H2PO4. The intermediate pH soil has divalent form, HPO4, and the alkaline, which has trivalent form. Excuse me. At 6.0 pH, both monovalent and divalent phosphate ions are present in the soil. Now, coming to the deficiency of phosphorus, there is development of anthocyanin, formation of necrotic dead spots on the lamina of leaf, petioles, and fruits. Decrease in protein synthesis, plants become stunted, root growth also gets restricted, distortion in leaf shape, slow maturation of root, the pith gets well developed, so therefore the xylem gets thin-walled and vascular tissues will be poorly developed. And we can also see premature leaf form. Now moving on to sulfur, the soil has sulfur in both organic and inorganic forms. It is taken up by the plants as in the form of SO4, the sulfate ions. Now here they are weakly absorbed. With the increase of soil pH, the absorption decreases. The organic sulfur becomes available to plants through biological oxidation as the microorganisms change it to sulfuric acid. Now the deficiency symptoms of sulfur are chlorosis, again which is the yellowing due to the degradation of chlorophyll, followed by anthocyanin development. Reduced growth with hard woody stem, extensive development of root system, decrease in stroma lamella, and increase in grana staking. Increase in starch and sucrose accumulation and the decrease in reducing sugars are also observed. Now, let us move on to potassium. It is present in the soil in the fixed, non-exchangeable form, exchangeable and soluble form. There is an equilibrium existing between these forms. It is absorbed by the plants in ionic form. It is the commonest free ion in the cell, which is found in abundance in the meristems. It plays a significant role in opening and closing of stomata. It also maintains differential permeability of cytomembranes and apical dominance. The deficiency symptoms of potassium are plant growth becomes stunted due to shortening of internodes, mottling and chlorosis first appear in older leaves, necrosis at the tip and margins of leaves, leaf tip curves, and destruction of pit cells of tomato and increased differentiation of phloem elements, decrease in carbohydrate metabolism, and increase in respiration. Now, let us move on to calcium. It is the cation available in fertile soils in plenty. A plant absorbs calcium from the soil in ionic form. The middle lamella is composed of calcium and magnesium pectate. It also influences the permeability characters of cell membrane. Calcium salt of lecithin is perhaps involved in the formation of cell membrane. Now, the deficiency symptoms of calcium are ultimate death of meristems, which are found in shoot, leaf, and root tips, chlorosis among the margin of young leaves. Later on, they become necrotic. Distortion in leaf shape, roots poorly developed, or may become gelatinous. Okay, and now let us move on to iron. The soil are generally not deficient in iron. It's available, however, depending upon the pH of the soil. Due to the increased soil pH, the plant begins to show iron deficiency symptoms. The plants take up iron frequently in ferric state, but the ferrous state is metabolically active. It is present in cytochromes and ferridoxins. Therefore, the symptoms or the deficiency symptoms of ions are extensive intervening and chlorosis in leaves. Only the young leaves show the symptom and not the mature ones. Inhibition of chloroplast formation due to inhibition of protein synthesis. Aerobic respiration is also severely affected when there is deficiency of iron which is seen. Now let us move on to the last major element of today's class, which is magnesium. It is present in soil in water-soluble form as silicates. It is made available to the plants in ionic form. It is an important component of chlorophyll and an important binding substrate for ribosomal subunits. It is also needed by carbon dioxide fixing enzymes, and its deficiency symptoms are intervenial chlorosis first seen in older leaves, development of anthocyanin, which is followed by appearance of necrotic spots, and depression of internal phloem and extensive development of chlorenchyma. Now let us move on to the minor elements. Now, the first minor element is manganese. It is an essential micronutrient to plants. It exists in soil in bivalent, trivalent, and tetravalent forms. However, plant absorbs this minerals in bivalent forms. In alkaline soil, due to oxidation, this element becomes unavailable to plants. Its deficiency symptoms are intervenial chlorosis followed by necrosis, chloroplast lose chlorophyll, starch grains becomes yellow-green, vacuolated, and finally degenerate. Green specks of oats, marsh spots of peas, and speckled yellow of sugar beets are all some of the symptoms which occurs due to the deficiency of manganese. Now moving on to boron. It was recognized by Warmington in the year 1923 to be the essential micronutrient to plants. It is absorbed by the plants as borate. If the soil pH is raised, it also becomes unavailable to plants like zinc and manganese. It is involved in the transport of carbohydrates. It indirectly participates in differentiation, nitrogen metabolism, fertilization, salt absorption, hormone metabolism, and water relation. So the deficiency symptoms of boron are death of shoot tips because boron is needed for DNA synthesis. Leaves develop thick coppery texture, then curve and become brittle. Root growth is arrested and flowers are not formed. Heart rot of sugar beet, internal core of apple, water core in turnip, and stem cracks of celery are some of the symptoms which occurs due to the deficiency of boron. Now moving on to zinc. It is absorbed by the plants in ionic form. Its availability decreases with rise of soil pH. It is needed by the plants in the biosynthesis of indole 3 acetic acid. And it also plays a significant role in protein synthesis. Therefore, the deficiency symptoms of zinc are intervenial chlorosis in mature leaves, beginning from margin and apex, followed by necrosis. Little leaf disease in several plants, rosette due to the shortening of internodes, white buds of maize, and mottle leaf in apple and walnut are some of the deficiency of zinc. Now moving on to copper. So, copper is an essential micronutrient to plants. The amount of copper found in soil solution is very low. It is taken up by the plant in ionic form. Like generally, besides, also present in protein and planktocyanin, which participates in electron transport chain and photosynthesis, the deficiency symptoms of copper generally includes necrosis at the leaf tip and margin, dieback disease, which is happening in citrus, and reclamation in cereals and legumes are some of the symptoms which happens due to the deficiency of copper. Now moving on to molybdenum. So here, it is first established as an essential micronutrient to plants by Arnold and Stout in the year 1939. It is present in soil as molybdate as well as in exchangeable and non-exchangeable form. The plant absorbs it in the form of molybdate ion and not as oxides. It is a component of nitrogenase and thus brings about fixation of gaseous nitrogen. The deficiency symptoms of molybdenum are chlorotic intervenal mottling of mature leaves, followed by necrosis and folding of leaves, inhibition of flowering or premature fall, white tail of cauliflower, which is the withering of lamina, and only the midrib persists as whip tail. And under severe shortage, mottling is also followed by wilting are all several common symptoms because of the deficiency of molybdenum. If coming to the last minor element or the micronutrient of today's class is chlorine. Now, it was added to be the essential micronutrient in the year 1954. Like manganese, it is also needed in Hill reaction. It helps in quick release of electrons from water. Its deficiency symptoms under aqueous cultures are chlorosis followed by necrosis and bronzing of leaves. Bromine may substitute for chlorine. Hence, with this, we are coming to the end of today's class. So besides all these, there are also other elements like sodium, silicon, aluminum, cobalt, gallium, selenium, and iodine, which also plays a major role in mineral nutrition. Hope this class has served useful to all. Thank you.